Dylan, do you have any idea what is about one carbon metabolism? You can write in chat, or even you can speak up if you want to. Anyone have any idea about one carbon metabolism? I just want to get what actually you understand when I mention about one carbon metabolisms, or you don't, or you can't relate to anything, any topic.
No, doctor.
Really?
Yes.
Okay. Take a look. Okay, I think I need to off my video ya sekejap because macam lagging sikit. Because I'm actually online from my home. Oh, first time hearing it. Okay, Adeline. Others, not sure. Okay. Okay, let's go to the next one. Okay, so this paper was published in 2017, 5 years ago, but actually there are more recent paper which is related to one carbon metabolism for sure. But I just take this for example, which is to relate the findings, whatever the findings that related to one carbon metabolism to health and disease. Okay, so one carbon metabolism basically is really close related to this the synthesis, okay, of the purines and thiamines, pyrimidines, and so on that relate to these formations of the DNA. Okay, but not just that, but one carbon metabolism is actually related to all metabolisms. Okay, all metabolisms includes the biosynthesis of lipid, biosynthesis of amino acids, okay, biosynthesis or degradations or metabolisms of fatty acids. Okay, so there are a lot of relationship to other pathway or other metabolisms that we can relate to these one carbon metabolisms. Okay, all right. So here, if you want to find out the paper, okay, this about this one carbon metabolisms, if you want to have a clear idea, not a clear idea, perhaps an overview about this one carbon metabolisms, so perhaps you can download this paper. Okay. Okay, so before we go into details, what is the one carbon metabolism? So perhaps I would like to introduce to you about the basic things. Okay, about the basic things that we are going to take a look into this when I give you more in details, or we go in depth about one carbon metabolisms. So basically, of course, we work a lot with the enzymes. Okay, so you know that enzymes it require no chemical groups for activity other than their own amino acid residues, but so meaning that the enzymes, it can actually works on its own, but it has its own rate. But then in some cases it requires an additional chemical components, which we call it as cofactor. Okay, either it can be in a form of organic or inorganic. Okay, so in organic form, it can be in a form of metals like iron, magnesium, manganese, and also zinc. And we have another types of cofactors as well, which is in the form of inorganic elements, which helps um, to give a boost to the catalysis of the enzyme. Okay. Okay, so about the okay, so the next one, and other than this, okay, I have one question for you. Do you understand what is the organic and inorganic elements? Do you have anyone knows what is the organic or inorganic elements? Come on, guys, wake up.
No.
Yeah. Okay, some said in the chat, organic elements that has carbon. Okay, organic element contains carbon. Lagi?
Organic element contain hydrogen, carbons, oxygens, and nitrogen, the basic things at atmosphere. And inorganic elements consist yang ni lah, metals, ions, semua tu lah.
All right. Okay, so organic elements, yeah, it contains carbon, but then we have the, when the first thing before other things, other elements actually exist in during the first time Earth formation. Okay, so we know that during that time, the only elements that we have a basic one is the CHO with the carbon, hydrogen, oxygen. Okay, so these three. So those elements or organic compounds, organic elements, so which should contain this CHO. Okay, so if you see any important enzymes or important synthesize or peptides that is very basic, so it's going to contain all these three elements CHO. Okay, so this is the organic elements, and other than that, we can consider as inorganic elements. Okay. All right, so the next one is about the coenzymes. Okay, previously we talked about the cofactor, and then this one is the coenzymes. So coenzymes, what is the coenzymes? Does it different from the cofactor? Okay, perhaps now you can Google since you are online. You can Google what is the difference between the coenzymes and cofactor, and try to take a look at it and think about it. Whether it makes sense. Okay. Perhaps you can find out the maybe a table that related to the difference between the cofactor and coenzymes and what are they.
Coenzyme organic, cofactor is organic.
Okay, who's speaking? Okay, maybe you can tell us what is the first difference between the coenzyme and cofactor.
My name is Nadira. Coenzyme is organic molecule, while cofactors is inorganic molecule.
Okay, cofactor is inorganic molecules. Coenzymes is organic molecules. Isn't it? Is that what you say?
Yes, doctor.
Okay. So, alright. So the next point, siapa yang nak bagi tahu? Okay, mungkin Muhammad Aslam, are you there? Perhaps you can tell us what is the other difference between coenzymes and cofactor?
Factor increases the speed of reaction while coenzymes acts as a carrier.
Okay. So cofactor can speed up the rate, yeah, and coenzyme act as the carrier to whatever that the specific atoms to other substrate or other enzymes. All right. Okay, so the next one, we have more actually, the difference between coenzyme and cofactor. Ooh, Nurin Liana? Nurin, are you there? Okay, Nurin, boleh buka kamera tak? Nak kenal lah Nurin ni mana. Boleh buka kamera? Actually, I think you need to on your camera while speaking. Tak boleh on video. Or okay, Nurin, what is your what can you share with us about the difference between cofactor and coenzymes? Mana Nurin ni?
I think her internet has problem, doctor.
Okay.
Sorry, I think the camera is a bit problem. It's lagging, I think.
Okay. Tan Xing yeah, who's speaking?
I was about to answer your question.
Okay. Nurin, okay.
So a cofactor is a non-protein chemical compound, while a coenzyme is a non-protein molecule. Okay, so different between
Sorry, again.
like a cofactor is a non-protein chemical compound, and the coenzyme is a non-protein molecule.
Non-protein molecule. Okay, non-protein molecule meaning that it's kalau dalam reactions of enzyme, perhaps it's actually a substance other than the substrate itself. Okay. Okay. Any.
So enzyme is an organic molecule, and the cofactor is an organic compound.
Okay. So other than that, anyone want to contribute to the answers? Okay, so no. Okay, let's continue. Okay, thank you all for your answers. Okay. Okay, sometimes some of the cases enzymes actually require both. Okay, both whether coenzymes. It actually require either one, whether it's the coenzymes or cofactor, or sometimes they require both. Okay, for the enzymes to function or to do the synthesis. So they need the coenzymes and more metal ions, which is a cofactor. Okay, cofactor for the activity. And we have actually a form of group of enzymes that we call as prostatic. So prostatic is when a coenzyme or the metal ion tightly or covalently attached to the enzymes. So this is when the coenzymes or any of the metal ion attached to this prostatic to the enzymes. Not the substrate, not at the substrate part, not at the substrate active site, but in other sites of the enzyme. So we can say that the enzyme is in the form of prostatic form. Okay, so so we have another form as well, which is holoenzymes and apoproteins, and I want you to find out what are these two. Okay, what are these two forms of holoenzymes and apoprotein? What are they? Okay. All right, so the next one is all right, so this is what we have discussed before. What are the difference between coenzymes and cofactor? So basically, coenzymes will provide they can act as the carrier to actually to carry a certain functional groups from one group of protein or one group of substrate to another types or to another groups of protein or enzymes or substrate. Okay. All right. So all right. Here is the what is the actually in the notes that I'm giving to you. I gave you it, but which is I already upload the notes in the bootstrap class. It's actually we have some corrections. So this here should be the coenzymes. So when you in every part which stated cofactor in this note should be coenzymes. Yeah. So sorry for that. I didn't correct that one in the notes. I just noticed it just now. So I'm sorry for that. So please correct in your notes. Okay, so please pay attention to this. So please replace the cofactor with the coenzymes. Okay, so the coenzymes functions here is in case of the one carbon metabolisms, the one that function to carry the one carbon from one group to another one is the coenzymes. So is coenzymes. So there is actually many parts. Okay, many parts that. Okay, so many parts of the process that involve one carbon metabolisms that we have discussed actually involved in the amino acid metabolism that we have discussed in previous topics, but I haven't discussed in details or specifically specific to that part. So perhaps you didn't notice about it. Okay, you didn't notice about it. But if you go back again to the process, to the step by step during the earlier part of the amino acids metabolisms till the end, there is actually some process, a bunch of it actually, which is involved these one carbon metabolisms. Okay, and so basically the three coenzymes that actively involved in the one carbon transfer is the biotin, okay, tetrahydrofolate, and adenosylmethionine. So these are three coenzymes that's actively involved in the one transfer, one carbon transfer. And so biotin is play a role to transfer carbon in its most oxidized states, which is the CO2. Okay, and then for tetrahydrofolate, it can transfer one group carbon in intermediate states and sometimes as methyl groups. Okay, and for S-adenosylmethionine, it can transfer the methyl groups, which is the most reduced states of the carbon. So these are three coenzymes and how they work to the transferring of the one carbon in the metabolism. Okay, so this is the biotin. Okay, this is a form of biotin, and actually biotin is a type of the vitamin, and even for the THF or tetrahydrofolate, all these, including the aromates, they are actually a form of vitamins. Okay, vitamin B. So which vitamins are this one, or are they? So biotin is vitamin B7. Okay, and what about these as adenosylmethionine and TH4? Okay, what types of vitamins are they? Cepat cari. Okay, biotin, what types of vitamins? B,
7.
B7. What about THF? This is a B, B9, B9. So please find out what is about the aromates. Okay, so the next one, okay. So the function of one carbon metabolism is to donate methyl groups to phospholipids, to biogenic amines, thymidine, and amino acid biosynthesis. Okay, so this is a function of one carbon metabolism. So that's why we can actually observe or find this one carbon metabolism in many pathways, and it's the location of the one carbon metabolism. It can happen mostly everywhere. Okay, most everywhere, and it's actually connected one carbon fragments in from serine, glycine, formate, and histidines. So mostly you're going to see the transferring of one carbon from serine, glycine, formate, and histidines. So it's kind of it makes a connection between from one intermediate to the next one. So from the previous example that I can say that, which is we have discussed about this, which is the degradation of the serine, right? So the serine it needs to be degrade into this glycine before it can go to the next step. So meaning that during the process, serine to glycines. So there is the process of this one carbon metabolism involved, right? And these are the like SAM, formyl THF, methyl THF, and this is methylene THF and methyl THF. So these are the other derivatives. Okay, there are the derivatives that actually will be produced in the presence of vitamins B2, B6, and B12. Okay, which is the play a role to actually transfer the carbon from other proteins, amino acids, or to the next intermediates. And these regulations is at individual enzymes level, meaning that they have their own, or they have their specific enzymes involved during the process. All right. So the next one is we take a look at this biotins, which is vitamin B7. Okay, vitamin B7, and so it is a types of vitamin B7. So this is B7. Okay, vitamin B7 called as biotin, and it's actually play a role in many carboxylation reactions. Okay, many carboxylation reactions. For example, to carboxylation of the pyruvate, okay, by the CO2 to form oxaloacetate. So we have discussed before on the TCA cycle, right? On the TCA cycle, how the process of this glucose to in the process of gluconeogenesis, there will be a process where pyruvate, a pyruvate will be converted into oxaloacetate, okay. Oxaloacetate. So here, during this formation of pyruvate to the oxaloacetates, there will be the process of carboxylation of the pyruvate, okay, by the CO2. Here, the biotin will act as a carrier to actually remove the part of the carboxyl group from this pyruvate into the oxaloacetate. Okay. Okay. So here we have the pyruvate into the oxaloacetate in the citric acid cycle. Okay. And how this pyruvate carboxylase is the main enzymes that involved for the to process the carboxylations that involve the CO2 to this formation of the oxaloacetate. And so still here we can relate to the amino acids metabolisms, amino acid metabolisms, because what happened is that from the pyruvate is actually we have many pathways that from the amino acid metabolism itself that can be end up with this pyruvate as the intermediate. Okay. So here is the descriptions or the explanation of this carboxylation of the pyruvate. So this carboxylation of the pyruvate, it proceeds in two steps. Okay, it proceeds in two steps. So this is the basically the structure, how the complex structure of the biotin. Okay, which is the actually carry the it can carry the the HCO2, which is a form of carbon dioxide from this carboxyl group, okay, from this carboxyl group of the pyruvate to this oxaloacetate. Okay, so here the complex of the structure, okay, of the component of the complex of the component of biotin, and then the pyruvate, and also the biotin. Okay, all right. So here you can see here we have the pyruvate carboxylase, which is play a role during the process of the transforming the pyruvate to the oxaloacetate. So what happened is that the biotin, okay, the biotin will actually attach to this lysine of the pyruvate carboxylase, and what happened is that the biotins the first thing is that at the catalytic site 1, if you can see here, we have two catalytic sites, which is the catalytic site 1 and catalytic site 2 at the pyruvate carboxylase. So what happened is that the biotins attach to these lysines of the pyruvate carboxylase. And what happened at the catalytic site 1 is the direct activations of this bicarbonate, okay, which is this carboxyl group will be released as the CO2, and the CO2 then will be attached to this the biotin, which is at this which is already attached to this lysines of this pyruvic carboxylase, and this is a form of carboxyl biotinyl enzymes, which is a complex of this biotin, the carboxylase, and also the part of the CO2 which already cleaved from this substrate. Okay, and what happens next is the oxaloacetate. Then it will transfer the biotin, which is in a form of activated CO2, to the second catalytic sites, which is what we have. So this is still at the first catalytic site here, but then later on the biotin will make an arrangement. Okay, will make structure arrangements, so then we can see how the CO2 now is on the second location or second catalytic site on the right. Okay, and then this where the pyruvic comes in. The pyruvic comes in, and there this pyruvic attached to this biotin here, and the CO2 will be and in the meantime the CO2 will be released, but then the CO2 will be attached to the carboxyl group of the pyruvic. So we finally get the oxaloacetate here. So oxaloacetate will attach to this functional groups of the oxaloacetate. Yeah. Okay. So that is the process of how the carboxylations of the pyruvic to the oxaloacetate with the presence of the biotin and also the pyruvic carboxylase as the enzymes. Okay. Right. So this is actually the same thing that comes from these figures. Okay, this just that to make it more simple. But then this one here that I showed you in the previous two slides, which is more detail on how the process of the transforming of from the first catalytic site to the second one by using the biotin. Okay, so this is just I think this one is much more simple process. Or simple steps from the formations of the carbonate, and then involve the pyruvic, and we get the oxaloacetate. All right, so that is the function of the biotin, and the next one we comes to folate. Okay, folate. Okay, folate is a form of vitamin B.
B9.
All right, B9. Okay, B9. So it's B9, and the reason why I put this avocado here, because B9 or folate actually the main source. We can get the B9 from the avocado. Okay, so avocado actually provides a lot of folate. So please, you can eat more folate. You can eat avocado a lot. Okay. So folate play a key role in the carbon metabolisms because it's essential for the biosynthesis of several compounds. And folate or B9 actually works together with the B12. Okay, works together with the B12, which is important for the formations of the very basic things of the formation of the DNA, right? Pyrimidines, purines, and all those things. You know that to the formation of the DNA or RNA, we need to have all those basic bases, right? Cytosine, ATCG or ATC. GU, uracils, which is in the RNA. So all those things actually need for folate for the formations of all these bases. All right. So in case of the folate, folate, which is a B9, it can be converted to this tetrahydrofolate by the enzyme we call the hydrofolate reductase. So so folate is actually in oxidized form, but the folate, it cannot be processed if it doesn't be in the reduced form. So meaning that in a way, folate need to be reduced in by the other components, or other elements, so that it can be activated. So it can be converted. In this case, it needs to be converted in 2 steps, which is tetrahydrofolate by the to tetrahydrofolate, which is in active form, by the enzyme of the hydrofolate reductase. So so this is the structure of this tetrahydrofolate, or you can actually write in this way H for folate, which is actually the same meaning to tetrahydrofolate. So this is the inner form of tetrahydrofolate, which is what we call as purine, which is only in bacteria, but it's actually a form of tetrahydrofolate. And this is the structure of the folic acid, as shown below, which is the this is actually a structure of folic or folic acid. And the folic acid or folate is reduced by the hydrofolate reductase in 2 steps. So the first thing is that the folate will involve this enzyme, which is the hydrofolate reductase to dehydrofolate. So the first thing is that it will become dehydrofolate, which is involve the double bonds at the N5 and N8 of the folate, N5 and N5 and N8. And then we get the hydrofolate. But then the second step is that they involve the transforming or chemical arrangements, which involve N6 and N7, and are reduced to tetrahydrofolate. So this is the first thing is that the folic acid or folate, and then they go through the process to the arrangement of the N5 and N8. Then we get in the presence of the hydrofolate reductase. Then and also we have the 2-NADPH as the cofactor in this case. So then we get this. Finally, we get this tetrahydrofolate. So the first step is to involve these N5 and N8 here, and then they involve the transforming this hydrogen to this N6 and N7. So basically, we're gonna get this tetrahydrofolate, and this is the structure of the tetrahydrofolate, which is complete after the 2 steps. After 2 steps of the reduction process from which involve the N5 and 8 and 6 and 7. So when we talk about this folate metabolic pathway, they actually involve a lot of this 1-carbon metabolism, and we can actually relate to these many steps that involve to these formations, to the formation of these amino acid metabolisms. So here you can see that we have the dietary sources that comes that the folate comes in the form of dietary sources. So these are the so this folate, it can go through the process of the reductions to this THF, which is the hydrofolate, and then to tetrahydrofolate, and then this tetrahydrofolate, then it can become activated. It is actually ready for many many pathways. It can do many works. It can give a key role to many metabolisms or pathway. So if you look at here, the tetrahydrofolate, it can go to these formations of the from serine to the glycines. Yes, and then it can be transformed, or it can be converted into another derivative, which is a form of N5 and 10-methyl THF towards the formations of this N5 and 10-methyl THF. So it can go in many ways from the just folate. Yeah. So actually, for informations, so we know that vitamin B. Okay, vitamin B is actually vitamin B and C. They are actually a form of water soluble vitamin, right? Water soluble vitamins. And we know that the vitamin A, D, E, and K is so it's soluble in the fatty acids, right? So this vitamin B and C, so it is actually it's soluble form. It can be absorbed. It can easily absorbed in water. Okay, so of course in the blood, and so it can then involve so many pathway in the metabolisms. All right, so if you look at here at the end of it, we have this formation of the N10 formyl THF. Okay, and this N10 formyl THF, it can go towards to this purine synthesis. Yeah, so this is the a very important process to these formations of the DNA and also RNA. Yeah, it's very important. So that's why women with the if you want, if someone's planning to get pregnant or in pregnancy, they have to take a lot of they have to get ready. They have to. It's better to get the source of the folate as the supplement. They should gain more folate so that that during the pregnancy, actually it's a process to it's developing new cells, new cells. So that's why we need a lot of folate. Okay, so it's very important. And here we have the at this side we have the 5-methyl THF. Okay, 5-methyl THF, where this 5-methyl THF, it can go back to produce the tetrahydrofolate, or it can also go towards these methionines, where this methionine it can go to another cycle, which involves the vitamin B12, which is the cobalamin. Yeah, the cobalamin is the B12, and so here we have this MS is the methyltransferase. So to do this, for this 5-methyl THF to get back to this tetrahydrofolate, because tetrahydrofolate perhaps it can go to either way, to these sites or to these other sites. So here they need this methyltransferase. Okay, methyltransferase is the enzymes that will carry this carbon, okay, to form back to this formation of the folate once again, and it can process to to any pathway in the cells. Okay, so about one carbon groups which carried by the folic acid or folate. So these are the form of the one carbon that can be carried out by the folate or folic acids, which is in the form of methyl, methylene, methanol, formimol, and also so all these group which actually are carried by the folic acid. And when this folic acid bring or carry a different parts of this one carbon, it will actually produce or become a different intermediates, or we call it as derivatives of the vitamin B. Vitamin B. So it can be vitamin. It can be in different form or different derivatives, which is related to this derivative of the vitamins B7, which is the folate. And okay, so the most reduced form of the cofactor carries a methyl group. Okay, it can carry the methyl group. Sorry, here again, this is coenzymes, not cofactor. Okay, so the most reduced form of the coenzymes carries the methyl group. So meaning that if, let's say, the folic acid bring or carry this methyl, it's actually the most reduced form of the coenzymes. Okay, so here we can assess to the reduction level of each of these methyl group. Okay, not methyl group. What we call derivatives, different form of derivative. So let's say the folic acids, attached to the methyl, then it's actually in most reduced form, which is we can say it's reduction level, which is 4, which is the highest one. And then if, let's say, the folic acids bring these carbon dioxide, then it's actually in the most low level of the reductions. Okay, and here the how the tetrahydrofolate, okay, the source of the active form of the folate or folic acid. If it's in a tetrahydrofolate, then it is actually ready for the transferring of the whatever the one carbon types, which is methyl or methanol, or so all these methyl, methanol, methylenes. So it can actually do the process. So in this case, the tetrahydrofolate, it can carry this one carbon that comes from the serine in the presence of the PLP, which is the so the PLP will help to carry the CH2OH, okay? And we get this glycine here, and there is the presence of the serine hydroxymethyltransferase, and over here we have the product, which is N5 and 10 methylene tetrahydrofolate. After the process of which is we get the side product as well, which is glycine, and then we have more the process. It can go to this formations of N5 methyl tetrahydrofolate, or it actually can go to this side, okay, which is to this formation of the N5 and 10 methylene tetrahydrofolate dehydrogenase, which is in the presence of this, and we get this N5 and 10 methylene tetrahydrofolate, and it can go to many parts or many pathway to this formation, to the formations of other derivatives of this folate. Okay, so it started with the tetrahydrofolate, but then it can go to it can produce this intermediate, and this intermediate here, it can go to 2 pathway towards this N5 methyl tetrahydrofolate, which is the where it attached to this CH3 red metal group. So we can say that N5 methyl tetrahydrofolate is most reduced form. And for this substrate, if it go towards down below, then it can go to this it can attach to this CH and H, okay, which is a type of femimino, okay, a type of amino, a form of amino. So this N5 amino tetrahydrofolate is the most oxidized state of these derivatives. Yeah. Okay. Okay, so yeah, what happened to the THF is actually we can relate to this one. Okay, we can relate to this pathway. Okay, so it can actually go to the during the process, it can actually process the transforming the serine to the glycines. Yeah? So we get the N5 enzyme methyl THF, and then we have the as I mentioned, it can go towards another pathway to get uh, the most oxidized states of the derivative, or it can get towards this way to the pathway of to get the form of the most oxidized states of the derivative of the folate. And so here, if we relate to what happened to these amino acid catabolisms to the one carbon metabolisms, so one of the examples that I can show you here is that the involvement of this transferring of the methyl group from the serine to the glycine, or from the glycine to the serine. It can go both ways actually. So and so here we can see the involvement of this transferring of the methyl group, yeah, from this glycine to the serine here, where we have N5 and 10 methylene H4folate, where it can transfer the methylene group to just be, and after that we get just H4folate. Yeah. And the next one is that the S-adenosyl methionines. So S-adenosyl methionines is a potential. It's a potential to transfer the methyl group from the hydrofolate. And okay, so S-adenosyl methionines, it play a role to synthesize the methionines to activate the methyl group cycle. If you look back to this cycle, yeah. So where does this methionines takes place, or adomate takes place, is to actually transfer the methionines, okay, to this formation of S-adenosyl methionine, or SAM. And here we have the activated methyl cycle. And the most important thing is that during the process, it involves the vitamin B12, yeah, or cobalamin. Okay, before we go into details, we go further into discussing about this adomate. Okay, adomate. So we take a break. Okay, we take a break. Okay, we take 5 lah. Okay, and later on I will continue with this one, adomate. Okay. Okay, class. Okay, let's continue. Sekejap saya share. Okay, so so if you can look at the look back to this cycle here. Okay, the cycle here. So we have what we have here is the form of 5-methyl THF, right? 5-methyl THF, which is after the process after a few steps towards the N5 and 10 methylene THF, and finally we get 5-methyl THF. And so up to this here, 5-methyl THF here, we need this methyltransferase. So this is the enzymes that will be responsible for the activations of this methionine cycle here. And what happens then? Okay, let's go back to this. Okay, let's go back here to these formations of the methionine and as adenosine methionine. So okay. So like I said, we have N5 methyl tetrahydrofolate over here, right? So we have the CH3, which is carried by the THF. Okay, THF. So basically, if let's say the THF is attached to this methyl group here, so this THF is not going to go anywhere. Okay, it's not going to go anywhere because we know that THF is very important. Okay, it's very important for the formations of the purine, pyrimidine, and all the DNA and RNA synthesis. Okay, RNA synthesis. But then, if let's say the methyl group is kept attached to this THF like this, so the THF cannot do this process. Okay, DNA and RNA synthesis. So what happens is that is going to be a pool of a complex of methyl THF. Okay, so that's what's going on. And so for say the THF needs to do the process of DNA and RNA synthesis, then what happens is that CH3 need to be cleaved off from this THF, and the THF is released and is free to do this process, okay, DNA and RNA synthesis. So how is the cleaving or the releasing of the methyl group takes place? So what happens is that we have the coenzymes B12. Okay, so B12. So what happens is that the B12 is going to help. Okay, is going to help the HF. So B12 is willing to carry the burden. Okay, is willing to carry the burden of carrying this CH3 methyl group from this THF to the coenzyme B12. And so this coenzymes B12. So we have the CH3 is going to be attached to this B12. Okay, B12. Okay, is going to take over from the THF. And what happened next is that for to the formation of the methionine. So what's next is that the B12 they need to take the homocysteine. So the B12 here they need to take the homocysteine, okay, and the homocysteine is going to give the CH3. Okay, so B12 is going to give the CH3 to this homocysteine to produce the methionine. Okay, to produce the methionines. So so over here we have the CH3 complex with the B12, and then B12 is going to give away the CH3 to this homocysteine, because homocysteine is on the way, the same pathway. So what happened is it offers the CH3 to the homocysteines. Then finally we get the formations of the methionine here, because the CH3 now attached to the homocysteine. Then we get the formation of the methionine. Okay, and so here methionines actually, they have this very powerful sulfur. They have powerful sulfur elements in the methionines. So what happens is that the sulfur, so it will react. Okay, it will interact or react with the ATP. So it will take over this phosphate group here. So this phosphate is going to be cleaved from this part of ATP, and then these methionines will attach to this ATP through the this sulfur, or we call it a sulfonium ion, which is a very powerful methylating agent. Okay, so so here as adenosine methionine is formed, and the sulfonium ion is very powerful, that attach tightly to the CH3. Okay, in the formation of S-adenosine methionine. And this is how the process goes. When the S-adenosine methionine is formed, then it can proceed to this formation of S-adenosine homocysteine, and homocysteines will go back to formation of the homocysteine. And this is where the process actually, the methionine it provides two important pathway to this to the process of the methionine. Okay, methionine, it actually have many fates as well. Not just towards this pathway to the formation of the THF to the DNA and RNA synthesis, not just only that, but the methionine, it can go through another pathway as well. Okay, and so meaning that over here, we know that B7, which is the THF, it's work together with the vitamin B12. Okay, vitamin B12. Not B7, B9. B9 is THF. B7 is biotin. Huh? So B9, it has to work together with the B12, because remember, B12 need to carry the CHF to release this THF from the methyl group, and it can carry forward to other synthesis. And okay, so B12, also known as cobalamin, and it is very important. And where does this B12 is produced actually? Anyone? Okay, so B9 and B12, since it's very important for the DNA and RNA synthesis. So if someone is deficient in to actually absorb B9 or B12, basically the disease that related to this B9 and B12 deficiency is like anemia or any disease related to the blood. Okay, and B12, for your information, B12 is produced or synthesized by gut bacteria. Okay, by gut bacteria. So those who have a problem with their gut system, which they cannot absorb the B12, So basically, this is going to be coupled with any blood disease like anemia or other disease. Okay, so this is a very important part of the B9 and B12. So hopefully, this information will help you to understand further about the process or about the role of these carbon metabolisms in the in many parts of the metabolism of the body. All right. Okay. Okay, just note. I think this is the note for you to understand. Okay, so this is about the vitamin B12 just now, which is cobalamin. Okay, which is work together with the B9. Okay, B9 which received this latter group from B9 and let the THF to go for the DNA and RNA synthesis. Okay, so this is the coenzyme B12, cobalamin. The structure of cobalamin, where it has the chlorine ring with cobalt in the middle, and which is the only metal in the molecule. Okay, it's actually a big molecule, but it just contains this cobalt in the middle of the chlorine ring, and which is coordinated by the nitrogen from 4 groups. Okay, 1, 2, 3, 4. And cobalt actually connected to several different types of ligands, okay, to form cyanocobalamin or methylcobalamin. Okay, so this is where the methyl group or whatever elements will be attached to. All right. Okay, so that's it for today's topic, about the 1-carbon metabolisms. Okay, anything that you would like to ask about the topic, 1-carbon metabolisms? We have another half an hour. What about the 3 speakers that is going to present for short and sweet session? Are you ready for presentation if we do it today, or you want to do it on Wednesday?
Wednesday, Doctor.
Wednesday, all right.
Yes.
So, or we will do it on Wednesday. All right, no problem. Okay. So hold on. We're not finished yet. Okay. Yes, hold on. Okay, for the attendance, you can actually record your own attendance ya dekat Putra Blast. I've created the link to the attendance today, so please go there and record your attendance from there. So in the meantime, do you have any questions related to this topic today? And for coming Wednesday, we're going to have presentations from the three speakers. So it's short presentation, yeah. So make it your presentations like within just 5 minutes. It's okay, yeah, 5 minutes. And I'm going to continue with the other topics on coming Wednesday. So if no questions, then I guess that's okay. No, yeah. This is what I want to mention actually. So you have the one assignment. Okay, one assignment. So I'm going to put it in the Petrol Blast later on. But what I need you to do is that it's about the assignments about vitamin B. Okay, vitamin B. So which is I just want you to explain or find the what are the contributions of this vitamin B, whatever vitamin B1 until vitamin B12. Okay, so what are the contributions to the metabolisms? Okay, to these metabolisms, to the TCA cycle. Okay, and so that is what I want you to discuss. Okay, about the relationship or relationship or the contributions of the vitamin B. Okay, vitamin B to these metabolisms. Okay, to the metabolism, especially to this TCA cycle. So I want you to find out where actually the this vitamin B, okay, whether it is vitamin B1, 2, 3, until 12, how and what are the contributions to the at which step or at which process of the TCA cycle they are actually contribute to the metabolism of the process. And we have vitamin B1, 2, 3, okay, 5, 6, 7, 9, and 12. Okay, so that is the proper 7 B1, 2, 3, 5, 6, 7, 9, and 10. Okay, what about vitamin B4 and 10? Okay, that one. Okay, for vitamin B4 and 10, we actually can synthesize it. Okay, our body can synthesize it. So it's not quite related to what we want to discuss about the TCA cycle and so on. But if you find out any information about B4 and 10, so you can include it as well. But the most important thing that I want you to discuss is about the vitamin B, source of vitamin B that we get from the diet. Okay, vitamin B1, 2, 3, 4, 2, 3, 5, 6, okay, 7, 9, and 12. So please find out what are the contributions. If you can relate to the process of amino acids and then relate to this process of the TCA cycle, then it's going to be good. Okay, so from there you can get the whole idea about the what we have learned, what we have learned from the earlier part, which is the amino acids, until the and compressed or include the part of the uncovered metabolisms, and then what we get is a very what we get is the we can relate to this process of the TCA cycle. All right. So I think that's all for today. So please don't forget about the attendance. So I'm going to upload the assignment in the Putra Plus, and also together with the due date and so on. All right. So if no more questions, and I'm going to end the session. Okay.
Doctor, when is our exam in week 5?
Yeah. One thing about the test one, your test one is going to be on the next week. Next week, Wednesday. Okay, next week, Wednesday. So, apa bulan ya? I think, I just mentioned to the group B kan. So, during the experiment practical session last time, I did mention to group B. So I'm going to repeat this information again. I'm going to mention it again. So your test one is going to be on the 20 April. Yes, that's correct. So it's going to be held on in the mal pengajaran 1 and 2. So you're going to take your exams face to face. Okay. So please get ready. But this week on Wednesday still we have lab practical 2 for group 1, and group B you're going to have your lab practical on the 27, which is another coming Wednesday, 27. All right. So that is my plan. So hopefully everything's going well. So get ready for your exam. Yeah? As I said, the topic that will be included in the test one is starting from beginning until amino acid catabolism. So and for this topic that we learned today, they have discussed this, so it will not be included. This one will be included in the test. Right. So yeah, that's it. Yeah, it's a 2 hours test. So we're going to start at 2. Okay, I'm going to ask you what time you want to start your exam. Which one is going to be convenient for you, 2 or 2:30?
2:30?
All right, 2:30. Okay, so 2:30. So jangan ada yang lambat ya, sebab kita dah start 2:30. So please get yourself ready. 2:30 dah ada dalam lab. Okay, and we're going to start the exam at 2:30. Right. Okay, so that's all for today. Any questions, you can just come to me lah. Okay, come to my office girl. Okay, so that's all. Then so thank you very much.
Format objective or subjective?
Semua ada. Rojak.
Okay, Doctor.
Okay.
Doctor,
Mm-hmm.
last question. About the essay, like, is it a 10-mark essay or is it a short essay?
I think it's going to be a long one.
Thank you, Doctor.
Okay.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.